to motivate the mouse to move, or he'll just sit there. And the goal, of course, of this maze is to get out of the water and go to a little platform that's under the lit top port. Now, mice are smart, so this mouse solves the maze eventually, but he does a brute force search. He's swimming down every avenue until he finally gets to the platform. So he's not using vision to do it. These different mice are different mutations that recapitulate different kinds of blindness that affect humans. And so we're being careful in trying to look at these different models so we come up with a generalized approach. So how are we going to solve this? Well, we're going to do exactly what we outlined in the previous slide. We're going to take these blue light photosensors and install them onto a layer of cells in the middle of the retina, in the back of the eye, and convert them into a camera. Just like installing solar cells all over those neurons to make them light sensitive. Light is converted to electricity on them. So this mouse was blind a couple weeks before this experiment and received one dose of this photosensitive molecule in a virus, and now you can see the animal can indeed avoid walls and go to this little platform and make cognitive use of its eyes again. And to point out the power of this, these animals are able to get to that platform just as fast as animals have seen their entire life. So this preclinical study, I think, bodes hope for the kinds of things we're hoping to do in the future. To close, I want to point out that we're also exploring new business models for this new field of neurotechnology. We're developing these tools, but we share them freely with hundreds of groups all over the world so that people can study and try to treat different disorders. And our hope is that by figuring out brain circuits at a level of abstraction that lets us repair them and engineer them, we can take some of these intractable disorders that I told you about earlier, practically none of which are cured, and in the 21st century, make them history. Thank you.
So some of this stuff is a little dense. But the implications of being able to control seizures or epilepsy with light instead of drugs and being able to target those specifically is a first step. The second thing that I think I heard you say is you can now control the brain in two colors, like an on-off switch.
That's right.
Which makes every impulse going through the brain a binary code.
Right. Yeah, so with blue light, we can drive information in the form of a 1, and by turning things off, it's more or less a 0. So our hope is to eventually build brain coprocessors that work with the brain so we can augment functions in people with disabilities.
And in theory, that means that as a mouse feels, smells, hears, touches, you can model it out as a string of 1s and 0s.
Sure. Yeah, we're hoping to use this as a way of testing what neural codes can drive certain behaviors and certain thoughts and certain feelings, and use it to understand more about the brain.
Does that mean that someday you could download memories and maybe upload them?
Well, it's something we're starting to work on very hard. Yeah, we're now working on some work where we're trying to tile the brain with recording elements too, so we can record information and then drive information back in, sort of computing what the brain needs in order to augment its information processing.
Well, that might change a couple of things. Thank you.
Thank you.